Hello, so my name's Andrew Wald, and my biology project this term is on epigenetics and schizophrenia. Schizophrenia, as a definition, is a chronic and severe mental disorder that affects how a person thinks, feels, and behaves. Some symptoms that are in this disease are hallucinations, all the way down to just a person showing no real pleasure in their lifestyle. So this is just a little introduction. Epigenetics is a study of potentially heritable changes in phenotype without changing the genotype. The DNA sequence is not altered, however, the active versus inactive genes are. And these are just which genes are expressed and show their function and which ones do not. The term epigenetics was derived from the Greek word epigenesis, which describes the influence that genetic processes have on development. The three main systems considered to initiate epigenetic change include DNA methylation, which is a huge topic, histone modification, and non-coding RNA associated gene silencing. The effect that environmental factors like stress and different lifestyles can impact the genome, which can influence the epigenetic change. And actually, a lot of research is being done on this, which is why the field of epigenetics is vastly growing. Much research is being done on the connection between epigenetics and schizophrenia. Schizophrenia, like I said earlier, is a major psychiatric disorder lacking unifying neuropathology. And this whole PowerPoint is based off this thesis statement right here. The research in the field of epigenetics has suggested that both an individual's gene, which is the nature aspect, and environmental factors, which is nurture or how something is brought up, can contribute to the onset of schizophrenia. Some studies suggest that schizophrenia can be linked to both genetic and non-genetic factors that, when combined, can result in the onset of this disorder. The non-genetic factors are epigenetic, like a modification of chromatin, which come from the DNA histone protein complex. And genetic is basically what it is, it's just heritable genes that come and go. So now we're going to get into the meat of this PowerPoint, and we're going to start off with epigenetic mechanisms, mainly the DNA methylation. But we're going to start by just describing what DNA is and how it forms. DNA is packed into the nucleus of the cells in the form of chromosomes. But before DNA is coiled into the chromosomes, it wraps itself around histone proteins to form loops, which are called nucleosomes. And these nucleosomes are the ones that stack up to form the chromatin, which then later on coil into the chromosomes. These genes are expressed based on how tightly or loosely the DNA is wrapped around the histone proteins. The coiling of DNA around the histone proteins is controlled by enzymes that bind methyl groups to the histones. When histones are methylated, the DNA is coiled tighter, causing transcription to decrease. So when transcription of certain genes is decreased, they're not expressed. So when transcription of certain genes are increased, those are the genes that are expressed, even though some of the genes that aren't being expressed are part of the genome. So just a little more. Other epigenetic modifications that can occur within the genome to influence gene expression are, like I said earlier, histone modification and non-coding RNA gene silencing. Histone modification is basically just an altering in the chromatin structure, which impacts gene expression. And the non-coding RNA associated gene silencing is a functional RNA molecule that is transcribed from DNA but actually not translated into any proteins at all. And DNA methylation is the most widely recognized, and studies show that it is a very potential explanation of the connection between environmental stressors and nurture and how schizophrenia is shown in an individual. So now we're going to get into that nature versus nurture aspect, specifically in schizophrenia, in two different topics, heritability and just the epigenetics of schizophrenia. So the heritability of schizophrenia, it actually runs in the family and is genetic, but it's not a completely genetic disorder. So that doesn't mean if it's prominent in your family, it doesn't mean you're 100% going to get it, but it's more likely you will. And actually, the risk of schizophrenia to first degree relatives, which is 9 times greater than the general population. It seems like a lot, but it's not it's not that significant. So now the epigenetics of schizophrenia. Environmental factors can lead to changes in the genome through DNA. And the environmental factors is basically how someone is brought up, like what goes around them, how they're treated, and stuff like that. So the DNA methylation, these genes that are involved in the neurological development and actual function slash cognition of these genes. Uh, until this day, studies continue to highlight that the postnatal, which is just after childbirth, after the postnatal caregiving environment has a direct influence on DNA methylation pattern of several genes. So the DNA methylation, the genes that are involved in the schizophrenia, based off this, they can be affected by the caregiving environment on an individual after the fact that they are born, how they're brought up into the world. So just a little connection now between these two, epigenetics and just the schizophrenia itself. Some studies that have been done to show this connection between the postnatal caregiving and just gene factors itself. So actually, there's been a few studies done, and some of the studies that have been done have been on twin siblings who actually have this disease or disorder. And one of them shows, specifically in this one of them shows it while the other one doesn't. And this research is just done to prove or show how one identical twin's genes is a little different than the other identical twins. And actually, it's been done on animals too, just like everything else in the world that's done on animals. We felt the need to do it here too. So many studies have been done on the identical twins because the whole identical part, which means they have complete genes that all look the same, but everyone knows that's not true because everyone's a little different. And actually, the genes in one twin sibling was a little different than the genes in the twin that didn't have it. And actually, that gene is what they've been looking at to find out how that modification has changed and how one twin has this disorder. And actually, on the rats, they use prenatal stress to induce schizophrenia behaviors inside the rat. These tests showed a significant increase in DNA meth transferase 1, which is in their cortex and the hippocampus, but not in the cerebellum, which is basically the main part of how someone would act or show certain things, because that's basically in the brain. So that's about it for this PowerPoint, but just a few closing remarks. They have new treatments and therapy that can be done. And they found this because of the connection between environmental factors and schizophrenia. And they have done more research on the antipsychotic treatments. And actually, one study specifically was to find out why patients that use this type of treatment have worked and why some have not worked. And actually, one study was trying to show which one was better, secondary generation antipsychotic drugs or the first generation ones. And this specific study shows that the second generation antipsychotic drugs are better than the first one. They said it was a little better because it had less side effects, but that was the main reason that I pulled out of the article. And that's about it for this PowerPoint. So thanks for listening.